During this webinar, we will talk about how we can interpret data from a qPCR experiment, the pretreatment we need to do, and the most common quantification strategies used for qPCR experiments. Finally, we will also talk about the usefulness of the MICE guidelines. We will start to talk about the baseline correction. To be able to compare samples in a qPCR experiment, they all need to start at the same level, and we normally adjust each amplification curve so that they start at the same zero baseline level. After correction, all amplification curves should have a flat baseline starting at the same level. This correction is normally done automatically by the instrument software. There are several ways that different software use to do the baseline adjustments. Normally more than one alternative is available in each software, and sometimes it can be useful to test different methods if it is difficult to achieve a good baseline with the default settings. Many software have an option where the software take a predefined number of cycles that they average and subtract from the amplification curve. Other options may be that the software identify where the increase in signal above the baseline level starts and take a number of cycles corresponding to, for example, cycle 3 to 2 cycles before the signal increase starts. This can be done either globally for the whole run or individually for each amplification curve. The first cycles of an experiment can sometimes show artifacts in the amplification curves, and you may want to exclude these in the baseline setting. And if the software does not exclude these automatically, you normally have an option to do so manually. Most software also include a slope correction function. This is to correct for the slope that is sometimes observed in the qPCR baseline, especially when using probe-based assays. It is, however, always important to inspect the baseline and especially the slope correction, so that these corrections have not introduced artifacts in the amplification curves. The next thing we need to do is to define the CQ values from the amplification curves. This can be done by setting a threshold level, and the CQ values are determined from where the amplification curve reaches the threshold level. Another alternative that is sometimes available is to use the maximum of the second derivative of the amplification curve and use that as CQ values. Most software have various algorithms to automatically set the threshold level, for example by using the noise of the baseline and setting the threshold level as a fixed number of standard deviation above the baseline, or by using a best fit of the data to a standard curve if such is available. When using automatic calling of the threshold level, it is very important to visually inspect where the software has placed the threshold level, so that it is within the logarithmic phase and above any artifacts from the noise of the baseline. The same criteria apply when setting the threshold level manually. It is normally easier to see where to set the threshold if using a logarithmic scale of the amplification curves. Make sure to place the threshold level in the logarithmic phase where the amplification curves are all linear and parallel, and above any artifacts from the noise in the baseline. As long as you are within this range, even though the actual CQ values change, the difference in CQ value between samples are the same, and samples in the same run can be compared with each other. Now we have our CQ values, and we want to use them to quantify the amount of target in our different samples. There are two main approaches to do this. The first is called absolute quantification. What do we mean by absolute quantification in real-time quantitative PCR? It is when we compare our unknown samples to a serial dilution of a standard of known concentration. The serial dilution is used to generate a standard curve, and from that we can determine the concentration of our unknown samples. Since we are always comparing our unknowns to a standard, the concentration we determine can of course not be better than the standard we are using. So, what is required of the standards? First, the standard curve should always be prepared to cover the entire range of our unknowns. We should never extrapolate outside the standard curve. This is always true when using standard curves, but perhaps even more so for qPCR, since we know that at the low concentration end we will get problems with stochastic variations and possibly primerdimers, and at the high enough DNA concentrations even DNA itself will inhibit PCR. Therefore, we only know the shape of the standard curve within the range we have actually tested. Second, to achieve the best precision in the concentration determination of the unknowns, we should also spread the standard points evenly over the studied range. When it comes to the composition of the standards, it can shortly be summarized in that the standards should be as similar as possible to the target we are analyzing. Preferably, we should use a DNA standard for a DNA target, an RNA standard for an RNA target, a linear standard for a linear target, single-stranded standards for single-stranded targets. We should use standards of the same length as the biological target. If the target is protein-bound or encapsulated, we should try to use standards of similar composition, and we should use a matrix for our standards that is as similar as possible to the one expected for our unknowns. However, when analyzing field samples, we often compromise on one or more of these points. So, what options are the most common to use as standards in qPCR? One commonly used option is to use a purified PCR product. The main advantage is that it's easy to produce and quantify, while it normally do not represent the field samples with regard to target length and sample matrix very well. Another option is to use a plasmid in which you have cloned your PCR product. It requires some more work to produce, but may be more similar in length to your field samples, at least if your target is a virus or plasmid or similar length product. If you are targeting genomic DNA, it is often possible to use genomic DNA as standard. The main advantage in this case is of course the similarity of the target length and composition between the standard and the target. However, the purification and stability may be a problem. If targeting RNA, especially if using a one-step reverse transcription qPCR, you need to use an RNA standard, or you need to be able to determine the reverse transcription yield some other way. The production is made by in vitro transcription of either a plasmid containing the T7 promoter with a cloned PCR product, or a PCR product with a T7 promoter added to the primer sequence. The main problem of using RNA standards is that the production is more laborious and that the quantification and stability of the standard may be a problem. No matter which option is used, the standard you have produced need to be thoroughly purified, quantified and diluted to cover the range of your unknown samples. The other way of quantifying the amount in samples is called relative quantification. Using this approach, we never determine the absolute amount in a specific sample, but only the ratio of the amount in one sample compared to the amount in another sample. This approach is mainly used in gene expression analysis. In relative quantification, we compare the expression of a gene of interest, a target gene, in different samples. But to be able to compare different samples, we normalize the expression in the samples, usually using a reference for each sample. Why do we need to do this normalization? Well, there may be several reasons. For example, different samples may contain different starting amounts. You may have different extraction yields for the different samples. The reverse transcription efficiency may be different for different samples. The degree of inhibition may differ between samples, and the degree of degradation of the DNA or RNA may be different for different samples. So how can we do this normalization? There are several options. Normally, you try to start with the same amount of material, but this may not always be easy, especially if you have small amounts of starting material. In some cases, it may be possible to count the cells and use the same number of cells for each sample. If total RNA is extracted and you have enough material without carrier, it is common to use the same amount of total RNA for each sample in the reverse transcription step. However, none of these methods take variation in RT yield or qPCR inhibition into account. This can to some degree be accounted for by using an RNA spike that is added to each sample and analyzed by a separate qPCR assay, or by analyzing the expression of reference genes that is constantly expressed in each sample in the study. In cases where a large number of genes are analyzed, the mean expression of all genes can sometimes be used to normalize the expression in the different samples. Even though normalization is often performed at different levels, the most common in gene expression analysis is to use one or preferably more than one reference gene. So what do we mean by a reference gene? Well, it's a gene that is constantly expressed in all samples in the study performed. However, there is no such thing as a universal reference gene. All genes are regulated under certain conditions, and using a regulated gene will actually distort your data, and you may draw the wrong conclusions from your experiments. Therefore, it is very important to verify that the gene you are using as reference gene in your particular study is not regulated under the conditions you study. It is also recommended to use more than one reference gene to improve the quality of the analysis, provided, of course, that all used reference genes are validated to be good reference gene for your setup. The normal way to select good reference gene is to run several potentially good reference genes on a representative set of your samples. It is important to include samples of all potential groups of data in your analysis. The data generated is then analyzed with an algorithm to identify the best reference gene or reference genes. The most commonly used algorithms are called gnorm and normfinder. Due to the way most algorithms select reference genes, it is also important to select genes that are not co-regulated when choosing candidate reference genes. Otherwise, co-regulated genes may accidentally show up as a preferred reference gene pair, even though they may be regulated in the sample studied. The result of reference gene selection will give you the best reference gene, and it's also possible to identify the number of reference genes that will give you the best precision in your analysis. Now, we have our reference genes, and we can calculate the relative expression of our target gene in the two samples. How this can be done depends on how much information we have and what we can assume about the qPCR efficiency for the different assays in the different samples. If all assays in the samples can be assumed to have the same efficiency, we can use the delta-delta-CQ equation. Here we assume 100% efficiency for all genes in all samples. If we can at least assume that each assay have the same efficiency in all samples, we need to take into account that the different assays may have different efficiencies. And if we cannot assume anything about the efficiencies, we need to work with the full equation and to actually determine the efficiency for each gene in every sample. Finally, we will talk about the MICE guidelines. So what is the purpose of these guidelines? Well, the aim is to provide specification on which steps that are needed to conduct RT-qPCR experiments properly, and also to provide guidelines to reviewers and editors to assure quality of published data. And why is that necessary? Well, real-time qPCR has in a short time become a popular analytical tool, and there have been a lack of consensus on how best to perform qPCR experiments. So, the MICE guidelines is a checklist to help ensure the integrity of the scientific literature, promote consistency between laboratories, and increase experimental transparency. So what challenges do we face when performing qPCR experiments? Well, the first challenge is sampling. When a sample is moved from its natural environment, it responds by activating some genes while repressing others. The measured profile may then not reflect the expression in the cell's natural environment, but rather the environment of the test tube. When samples are preserved for later analysis, the mode of preservation may compromise the quality of the RNA, introducing bias. Old samples may have compromised RNA due to degradation. RNAs, particularly different types of RNAs, extract in different yields, which may complicate comparison. The reverse transcription yield depends on priming strategy, the transcriptase used, and the reaction conditions in a gene-specific way. The variation can be 200-fold. Different RNAs amplify with different yields due to the folding of the template and the assay performance. When measuring expression, normalization is important for comparative analysis. As we have seen, several designs and normalization options are available. So what is needed to overcome the things that may have been overlooked before? First, we need to make sure that all steps in the procedure are described in more detail, and that the nucleic acid targets are thoroughly analyzed, that the qPCR assays used have been properly validated, and that information is provided about how data analysis is performed and the choice of reference genes. To simplify this, 9 different areas have been identified, with items that should be controlled, going from experimental design to data analysis. Each area have different numbers of items to be checked, and there are both items that are essential, that must be submitted with the manuscript, and items that are desired, that should be submitted if available. These points are summarized in a checklist that is included in the paper, where essential information are marked with an E and desired information with D. That can be followed to make sure all required information are provided. If you want to read more about the MICE guidelines, you can do so in this publication and its later revisions.